Okay, welcome everybody to Wednesday Compass Seminar. Today we have Shannon Doherty. She's a fifth-year PhD student with plans to graduate this year, and she works with Dr. Hillary Close in the OCE department. She obtained a BS in geology from Allegheny College and has won multiple awards and has a publication in Review. And last year she won second place in Best Student Seminar. So today should be a good seminar and I'm glad everybody is here. So today her talk is called Towards Quantifying Zooplankton Fecal Pellet Contributions to the Biological Pump. So very important stuff, carbon cycle. Take it away, Shannon.
Thanks, Anne. I'm going to share my screen. Okay. All right. All right, so thank you, Anne. As Anne said, I'm going to be talking about moving towards quantifying zooplankton fecal pellet contributions to the biological pump. And I'm excited to have you all think about fecal pellets for a little while, probably more than you ever have, unless you've been to another one of my talks. So happy St. Patrick's Day. To go through the outline of this talk, I'm going to introduce why we are interested in zooplankton fecal pellets in the biological pump. I'll lay out the problem of how we assess zooplankton fecal pellet contribution to particulate organic matter. I'm going to split my results into two parts. The first will be improving the fecal pellet quantification method in particles with stable isotopes. The second will be applying our new tool to a water column, which is Monterey Bay. And then I'll talk about the future work that we're going to do to address the remaining questions associated with this method. And as a bonus, I'll talk about something cool that we found from our study to quantify fecal pellets that also might help think about fecal pellets in the biological pump. So to begin, why are we interested in zooplankton fecal pellets in particulate organic matter? To start, I'm going to go over some definitions, and the first is zooplankton, which are really central to this talk. So zooplankton are animals that are weakly swimming and therefore transported by currents or drifting, and they're typically 0.2 to 5 millimeters. There are a lot of zooplankton outside of that size range, but that's the majority of what we're looking at. And here are some examples of zooplankton. We have our crustaceans like krill and copepods. Copepods are really important zooplankton group, and amphipods here. We also have gelatinous zooplankton like salps here and annelid worms here, and these are important because they're anticipated to become more prevalent as the ocean warms. We have jellies, so these would definitely fall outside our 0.2 to 5 millimeter range. And we also have larval marine organisms like larval fish. Some more definitions. I'm going to talk about dissolved inorganic carbon, which is the dissolved carbon dioxide gas and carbonate ions in seawater. Particulate organic carbon are organic molecules that make up particles. And that's anything that's greater than 0.5 microns, so anything that would collect on a 0.5 micron filter. I'll talk about particulate organic matter, which includes particulate organic carbon, but also nitrogen, oxygen, and sulfur, and the other elements that make up organic matter. So to begin, we'll go over the open ocean carbon storage mechanism that we call the biological or the soft tissue pump. So in the atmosphere, we have carbon dioxide, which is equilibrating with the mixed layer and forming dissolved inorganic carbon. This dissolved inorganic carbon is fixed by photosynthesis, which is done in phytoplankton, and it converts dissolved inorganic carbon to particulate organic carbon, which is proteins and sugars and everything that makes up life. Then this particulate organic carbon is consumed by animals and bacteria and respired as dissolved inorganic carbon. And then this dissolved inorganic carbon is back into equilibration with the atmosphere. And this is the ultimate fate of all particulate organic carbon. However, because we're in the ocean, these particles are also sinking. And as they sink, they're being respired and turned back into dissolved inorganic carbon. but the depth at which this respiration occurs or remineralization occurs controls how long that carbon is stored in the ocean. So the deeper the carbon makes it before it's remineralized, the longer it will remain in the ocean. And I'm particularly interested in this component of the biological pump. What makes up these sinking particles and what controls how deep they go before they're remineralized in the ocean. And to explain how zooplankton factor into this sinking particle export, we'll talk about Stokes Settling Law. So this is tells us the velocity of a particle is proportional to the density of the particle, times the radius of the particle squared. So the larger and the denser the particle, the faster it will sink, and the faster a particle sinks, the more likely it will be stored for longer in the ocean. So over here we see zooplankton consuming phytoplankton and eliminating phytoplankton as fecal pellets. So zooplankton aggregates small particles by feeding on phytoplankton and detritus, and they eliminate them as dense fecal pellets. So zooplankton are transforming organic matter that's less dense and smaller into denser and larger particles. And these fecal pellets may be disaggregated into small particles at depth and remineralized and returned to the dissolved inorganic carbon pool. So over here, I starred this one mechanism that I just talked about. This is a more complex cartoon of the biological pump and its interactions with organisms. But zooplankton also have other interesting feedbacks on the particle cycle. So the first is zooplankton vertical migration. And this is a mechanism by which zooplankton physically move carbon from the surface where they feed at night to depth where they rest during the day. So they're also physically moving these particles. We also have zooplankton that are living below the euphotic zone, and those are consuming particles and repackaging them, but they're also breaking up particles because they're very messy eaters. So they're doing both things at the same time. And to bring us back to why we're interested in this more generally, zooplankton are important in determining how climate change will impact ocean carbon storage. So this is a really messy flowchart of how warming in the atmosphere and ocean can lead to either increased carbon storage or decreased carbon storage or a mixture in the biological pump. And I'm going to focus on this very bottom path where reduced coverage in the Antarctic leads to declines in ice, diatoms, and krill, and increases in small phytoplankton and salps. This would result in reduced krill fecal pellets, which would lead to decreased carbon storage, and increased salp fecal pellets, which will lead to increased carbon storage. So one can imagine that being able to quantify how much these fecal pellets are involved in this biological pump would be essential to testing these hypotheses about how the biological pump will change as the ocean warms. So to summarize, assessing the contribution of fecal pellets to particles is essential to understanding the role of zooplankton in the biological pump. And our current estimates of fecal pellet contribution to particles can range from 0 to 100%, so the entire spectrum, and they are regionally, seasonally, and temporally variable. So here on the right, we have a picture of a filter that has our organic particles on it, and our question is, what is this? How much of this is fecal pellets, and what are the other components of it? And to solve that problem, we have to assess how zooplankton fecal pellets contribute to particulate organic matter. So we have two current methods. One is visual assessment from traps. So in this method, sediment traps are deployed and they collect particles, and then these particles are imaged and identified as fecal pellets. And then we can measure the size of these particles and calculate a biovolume and convert that biovolume into a carbon measurement. So there's a certain calculation for carbon to biovolume for fecal pellets. And the pros of this method is it's quantitative. You get a number out of this, but it's biased to intact and recognizable fecal pellets. So a human or a computer that's trained by a human has to be able to recognize these fecal pellets in the first place. So if they're no longer morphologically distinct because they've been chewed up or broken up, then we won't be able to recognize them with this method. Our second method is using biomarkers. So these are chemical compounds that are associated with zooplankton, in this case fatty acids. And we can measure which fatty acids here are associated with zooplankton and compare those to other fatty acids associated with other organisms or organic matter states. And the pros of this kind of method are it's not biased to identifiable fecal pellets, but unfortunately it's qualitative. And these biomarkers are derived from zooplankton biomass, which means they may or may not be in fecal pellets. And the extent to which that they're in fecal pellets is probably very variable and not easy to measure. So our ideal method would be one that's quantitative, that's not visually biased, and is proven to separate fecal pellets from other sources of organic matter. And this last one is the real challenge because fecal pellets are made of other sources of organic matter. So we're trying to separate fecal pellets from what they're made out of, which is a very complicated situation. So as an isotope chemist, my solution to this is considering isotopic fractionation during digestion. So digestion will impart a fractionation or an isotope reaction of the nitrogen isotopes onto a particle when a zooplankton consumes it and digests it. And to explain this a little more, I'll go through what isotope fractionation looks like. So if we have a chemical reaction, we have our reactant, which is made up of a mix of the heavy or rare isotopes in these darker red circles, and the light common isotope, which is made up of the lighter circles. And when this reactant undergoes a chemical reaction that doesn't run to completion, we have more of this heavy isotope left in the reactant because the heavy isotope requires more energy to move into the product. So we get in the end is a reactant that has a higher ratio of heavy to light isotope, in this case 3 to 5, and a product that has a lower ratio of heavy to light isotopes, which in this case is 1 to 3. And when we measure isotopes, we use delta and 15 values in the case of nitrogen, and this is just the ratio of nitrogen 15 to nitrogen 14. So this would result in a reactant having high delta and 15 values and a product that would have low delta and 15 values. And I use amino acids in particular, so I measure these isotope ratios in amino acids. And we do this because amino acids are ubiquitous in all organisms, and the same 20 compounds, the same 20 amino acids are used in all proteins as building blocks. Amino acids also contain both carbon and nitrogen, so I'll talk about nitrogen isotopes in this talk, but we can also measure carbon isotopes on the same amino acids. And these amino acid isotope ratios can be used as signatures. So to kind of illustrate what I mean, I have the citric acid cycle on the right. We have our reactants in black and our amino acid products in red. So just like before when I was talking about the isotope fractionation with reactant to product, that happens each time amino acids branch off of this cycle. So each of these amino acids will have its own distinct isotope value based on where it branches off in the cycle. And it happens, these reactions go to happen with various rates and to various extents in different organisms. So all of these amino acids will have isotope ratios that are distinct to a certain metabolism. So to put this all together, how we are going to quantify zooplankton fecal pellet contribution. First, we're going to isolate fecal pellets. That's important. We need our fecal pellets to measure. Then we're going to isotopically characterize the fecal pellets. Then we will determine an isotope signature for fecal pellets, which must be ubiquitous to zooplankton fecal pellets, but only fecal pellets. Distinguish fecal pellets from other organic matter and POM, such as phytoplankton, microbes, etc. And then we're going to test this method on particulate organic matter, so this also must be measurable in our particulate organic matter samples. We'll jump right into quantifying fecal pellets using stable isotopes. And To satisfy the first requirement that I just discussed for our signature, we need to have a signature that's ubiquitous, so it's not distinct to one organism or region. And to do that, we have 4 different locations that we collected various zooplankton species. This is a map where the color bar is the annual average chlorophyll, which will be kind of a proxy for your nutrients and production in any given region. And our first cruise, this is the NASA Exports project in the Northeast Pacific. We collected Salpa aspera and Vibilia propinqua. So these are those jelly salps that act as vacuums and suck up particles and produce dense fecal pellets. And they were very abundant during this cruise and a very important player in the zooplankton available at that time. And then we have Vibilia propinqua, which are parasites of salps. So they're living inside the salps and consuming both salp tissue and the particles that the salps are collecting. So they're an interesting end member to study. And then we have 2 cruises from Bermuda in the Sargasso Sea, and this is an oligotrophic subtropical gyre. So we have low nutrient concentration and low production overall annually. And from here we have another set of salps, and we have the mixed community of zooplankton. And we measure the mixed community here for 2 reasons. One is that in the Sargasso Sea, there's no one zooplankton species that predominates, so you couldn't pick one species and say that this one is the most important here, the most indicative of zooplankton communities. The second is anywhere when you measure your mixed community, this is really more representative of what you'd get overall in your water column because you have everyone, all of the players there. And as a contrast to that, in the Southern Ocean, where there's really high production seasonally with the ice melt and the ice edge diatom blooms, we have the Euphausia superba, which is the Southern Ocean krill, and this really is the most important zooplankton species in the Southern Ocean. It forms the base of the animal food web. So when we think about whales consuming krill in the Southern Ocean, this is that krill that we're thinking about. So we measured all these species. We collected their fecal pellets and measured their zooplankton biomass as well. And just to go through how we get fecal pellets from live zooplankton, we do have to incubate them using specially designed containers for this. So each container will have an interior container, that's this kind of orange one, that will have a mesh bottom. And then we'll have an exterior container that's intact. We'll fill this with filtered seawater. We'll incubate the zooplankton above the mesh and let them do their thing. And the fecal pellets will fall through the mesh and collect in the bottom of the container. And we pick a mesh specifically such that it's smaller than the zooplankton biomass, but larger than what we would expect the fecal pellet diameter to be. And at the end of the incubation, we can separate these 2 containers, and we'll have an external container that contains seawater and fecal pellets, and we'll have that interior container where zooplankton have collected onto the mesh of the internal container. So I'll filter the fecal pellets out of this, and then I will analyze them. So first we extract the amino acids, then we purify them so we know we have a solution of only amino acids. Then we run stable isotope analysis on these amino acids with our isotope ratio mass spectrometer. And what we end up with is isotope ratios of 14 amino acids. So we'll have a chromatogram that looks like this, and each peak is an amino acid. And then for each peak, we also have an isotope ratio that's measured. And then once we have those isotope ratios, I can run statistical analysis. In this case, I did ANOVA and Tukey tests to determine the distinction capabilities of each of these amino acids on our end member groups. And these end member groups have to be things that are also in particles. So in order to differentiate fecal pellets from other things, we have to have those other things too. So I didn't measure all of these, and we needed to collect data from the literature. And the data from the literature needs to both be nitrogen isotopes of amino acids specifically, but also overlap with the amino acids that I measure. And we were able to find some of those. So our literature data for phytoplankton, we have 12 samples of phytoplankton. our microbially degraded detritus. We have 4 samples of microbially degraded detritus. So I'm going to highlight this as somewhere that we need to make more measurements as an isotope community. Similarly, live bacterial cells that we would find in organic matter, we have no data that overlapped with those. Our fecal pellets, we had 7 samples, and a combination of literature data and our data give us 56 zooplankton samples. This is a really well sampled end member. So we're going to take these end members and see which amino acids can reliably differentiate fecal pellets from each one of these other end members. So here are the results from the statistics. This is the amino acid here at the top in bold, and then each end member in a box. And if there's a green line connecting the end member to one another, there was a statistically significant difference using this amino acid. And then a red dotted line is a statistically indistinct end member pair using this amino acid. So there were 4 amino acids that were the most successful at separating these end members. On the left-hand side, we have threonine and glutamic acid. These were the only 2 that were successful in separating microbially degraded organic matter from fecal pellets. And on the right-hand side, we have alanine and leucine, which were the only 2 that were successful at separating microbially degraded organic matter from phytoplankton. Now, we could use all 4, but we have some suspicions that the zooplankton statistics is due to, statistical result is due to a trophic fractionation effect that we know about. So we're going to choose threonine, which is really well established as correlated with animal digestion. And on the right-hand side, we're similarly going to pick alanine, which is really well established as correlated with microbial digestion. And we're not quite sure what the underlying reason is that the leucine was capable of separating them. So these are both theoretically supported amino acids to use, and together they will separate all of our end members from one another. And we can plot that separation. So here we have threonine on the x-axis. It's normalized to phenylalanine, which is just to remove any isotope effects due to location. So we can compare samples from different global regions to one another using this normalization. And on the y-axis, we have alanine that's similarly normalized, the delta N15 value of alanine. And all of our samples are plotted on here in dots. So we have zooplankton in the blue here, dots, and then the mean is in the diamond with standard deviation error bars. Fecal pellets in brown, microbially degraded organic matter in this orangey yellow, and phytoplankton in green. And we can see that this does a good job separating these end members from one another, and we can use these 2 to pull apart fecal pellets from other organic matter end members in the water column. And now that we've generated this tool, we need to test how well this works on an actual water column. So in this case, we're going to test it on Monterey Bay. We collected Monterey Bay particles with some researchers from Embari and UC Santa Cruz, and we put down pump systems that filter particles out of the seawater in situ. So the seawater gets pulled through a series of filters here by the pump and then pushed out at the bottom. And we used a stack of 3 different filters to get 3 different size classes. So our largest is greater than 100 microns, our intermediate is 20 to 100 microns, and our small particles are 0.7 to 20 microns. And just to put this into perspective, our human hair is about 20 to 70 microns wide, and our human visibility threshold is about 40 microns. So these small particles, we cannot see individual particles with our naked eye. Some of the intermediate and large particles we can see with our naked eye, but we're looking at the scale of like a human hair. So very, very small still. And we measured these 3 different particle size classes at 10 depths. We started at around 5 meters and measured pretty thoroughly through the euphotic zone, which ends in Monterey Bay at this time around 55 meters. And then below the euphotic zone, down to 500 meters. Our total water depth here was 900 meters, so we avoided any interactions with the sediments here. So we measure these particles and we can get in nitrogen concentration in micrograms per liter. This is a logarithmic scale on the x-axis of nitrogen concentration in micrograms per liter, and depth on the y-axis. And from this we can see that the majority of our particles, at least in concentration, are the smallest particles. And this is pretty typical of a water column. We see this everywhere globally. And we have less of our intermediate and large particles. However, our intermediate and large particles are the particles that are sinking. So they're very important, even though below the euphotic zone, which is this dotted line, they have a much lower concentration. And that's because the sinking speed of particles that are this small is almost negligible. So we consider them suspended particles. And again, we have these particles now, and our question is, what is in them and how much of them are fecal pellets? So to set our expectations for that, we can plot our particle size classes over this categorization of particles, things that you would find in particles in the water column. This is another logarithmic scale on the x-axis. And our sampling techniques are designed such that we exclude zooplankton, and we do quality control to make sure we have no zooplankton biomass in our samples. So I'm going to change zooplankton here to fecal pellets because the feces of any animal would be on the same relative scale as their biomass. So we're going to change that to fecal pellets. So to overlay our large particles, what falls in this group is most of our fecal pellets, our largest phytoplankton, and large aggregates. Our intermediate particles will be our small fecal pellets, known as mini pellets, and most eukaryotic phytoplankton and medium aggregates. And our smallest particles will be most prokaryotes, so that would be our smallest phytoplankton and our heterotrophic bacteria, as well as detrital particles. And detrital particles can be similar in composition to these two larger size classes because anything in the larger size classes can be broken up and put into the smaller size class. So we can have disaggregation happening, moving things from the larger size classes to the smaller. So now that we know that these are probable components in our fecal particle samples, we can overlay them onto this classification scheme that we developed in the last part of this talk with threonine on the x-axis and alanine on the y-axis and our end members. So when we add our particles in these dark blue star symbols, we see that they really do fall in between our defined end members, particularly microbially degraded organic matter, fecal pellets, and phytoplankton. So this is applicable to these particles. I'm going to go through them qualitatively, size class by size class, to talk about how this matches our expectations and some interesting things that we can find out of this. So just generally, what we would expect is that in the euphotic zone where phytoplankton are living and photosynthesizing, we'd have a lot of phytoplankton signature. Below the euphotic zone, phytoplankton are no longer photosynthesizing and they're being consumed by microbes, so we might expect more microbially degraded organic matter, and that'll happen throughout the mesopelagic. And then fecal pellets could really be anywhere in this water column where zooplankton are living. We're most interested in where fecal pellets show up in the mesopelagic because that's where they will be contributing to carbon storage. So if we go through our large particles, our first particle at 27 meters looks a lot like phytoplankton. This matches what we would expect. Similarly, our particles below the euphotic zone look like degraded stuff. So that's similar to what we would expect. And then we see that our fecal pellets show up around 200 meters and they are present all the way down to 500 meters. So we can see a fecal pellet signature in our particles in the mesopelagic, which is great. Our intermediate particles, we would expect similar things out of those. So interestingly, at 55 meters, looks like we have a particle that's dead center in between the fecal pellet end member and the phytoplankton end member. We have below the euphotic zone degraded stuff similar to our large particles. And then in the mesopelagic, again, we have fecal pellets, also similar to our large particles. Our small particles are a little different. So at the very surface, we have a very strong phytoplankton signature, but at the base of the euphotic zone and a little below the euphotic zone, we have a combination of what looks like fecal pellets and phytoplankton, which is interesting. From about 100 meters to 260 meters, we have degraded stuff, a little bit similar to our larger particle size classes. And then at 355 meters, we have what resembles fecal pellets. And at 516 meters, we have what might be fecal pellets or a combination of phytoplankton and fecal pellets. So this is all really interesting, but this is all very qualitative still. And these are obviously all mixtures of these three end members put together. So we want to be able to take this and make it quantitative. And to do that, I use a tool that's really popular in stable isotope ecology, Bayesian mixing model, which we can apply to an isotope biplot. So here we have a biplot really similar to the one I just showed you. We have delta N15 on the y-axis and delta C13 on the x-axis, but these could be any two isotopic variables. We have our end members and our potential diet. So these are animals, and here we can think about you are what you eat. So these animals are a mixture of the end members of their diet. We have end members with error, and then we have our mixture animals that fall somewhere in between all of these end members. And we can run all of this data through a Bayesian model. And what we get is a proportion of an end member, in this case grass, to each of our animals. So we can quantify how much grass is contributing to each animal and the error associated with that estimate using this model. So I'm going to do that with my particles. Here's a reminder of how similar these two plots look. And because we know that our sampling techniques exclude zooplankton biomass, and our quality control techniques also exclude zooplankton biomass, and our particles don't really resemble zooplankton biomass, I'm going to go ahead and eliminate that as a potential end member to these particles and focus on fecal pellets, microbially degraded organic matter, and phytoplankton. So when I run this data through a Bayesian mixing model, I can get a plot like this, which is the percent of particle pool that comes from fecal pellets for each of our particle size classes with depth. So I want to pause here and be like, yay, this is what we wanted. We wanted to be able to quantify fecal pellets in our different particles. And it looks like we can do that reasonably well here, and we can associate an error with this estimate. I also want to point out that there's a non-negligible contribution of fecal pellets to our small particles. And this is something we would miss if we relied on that the visual method that would exclude any broken up fecal pellets or detrital fecal pellets. So that's really interesting as well. And we can also see that these contributions vary with the depth, and the depth at which fecal pellets show up or contribute a lot might have something to do with the diel vertical migration of zooplankton or the sinking speeds of these particles. So there are a lot of ways to interpret how these are variable and how they change with depth. So this is really cool. This is very interesting, and this was the goal of this entire study. So I can zoom in and talk about a little bit what we can take from this in Monterey Bay. So here I have the same plot I just showed you, which is the percent of the particle pool from fecal pellets with depth. I just took our larger particles because these are the ones that are sinking. So these are the ones that we're thinking about contributing to carbon export. And a similar plot is over here with particulate nitrogen concentration on the x-axis. And I started these plots around 100 meters, which is our export depth. So this is the depth below which we would imagine that these particles are contributing to carbon storage in the ocean. So that's why I start the plots there. So first, we can quantify that our fecal pellets are contributing 40 to 60% of our sinking particles. And this contribution is variable with depth, which is likely due to migration and sinking, like I just said on the previous slide. We can also conclude, because 40 to 60 is a pretty large portion of our particle pool, that zooplankton are playing a major role in the biological pump in Monterey Bay. Now, to add to that, I use this estimate of the proportion of particles represented in, the proportion of fecal pellets represented in particles to the nitrogen concentration of these particles. So these open boxes are the total nitrogen concentration that fecal pellets are responsible for. And then the stars are the total nitrogen concentration in these particles overall. And what we gather from this is that, yes, it is variable with the depth, but generally these two size classes contribute similar particulate nitrogen in their fecal pellets. So you can see that these concentrations are relatively similar. And that's despite having very different concentrations with the depth between the two particles overall. So what I gather from this is I can hypothesize that small and large zooplankton are contributing to export equally here. Both smaller zooplankton in these 20 to 100 micron fecal pellets are contributing a similar amount total to these greater than 100 micron fecal pellets with depth. So this is an example of some interesting interpretations that we can take away from a study like this. And we can also apply this to our particle traps and other particle collection techniques. And in fact, this is what the other students in the lab are doing. So a summary of our fecal pellet quantification results from this study. We can distinguish fecal pellets from other components of organic matter using the two amino acids, alanine and threonine delta and 15 values. Bayesian models can allow us to quantify fecal pellet contributions and provide an error associated with that estimate. We can apply this quantification to our measurements of particle concentrations and get an amount of fecal pellet carbon and nitrogen estimate for the water column, which is also amazing. However, we need to characterize the microbial end member much better, which is what I mentioned back when I introduced the end members. And the isotopic signature of the activity of microbes on fecal pellets is also unknown. And I'm going to go through that a little more in the next few slides. But first, I want to mention that the fecal pellet quantification method is under review at Limnology and Oceanography, and our Monterey Bay study is in prep for GCA. So our future work and remaining questions are mainly, do fecal pellets retain their signature even after microbial degradation? So as fecal pellets are sinking through the water column, they're still being chewed up by microbes, and this might have an effect on their isotopic signature that we haven't characterized. And I have two major hypotheses for how that could go. The first is a fecal pellet end member could have its signature entirely replaced by the microbes that are eating it, and in this case would just move from the fecal pellet end member space into the microbially degraded end member space. We could also have a fecal pellet that retains its threonine signature, but because alanine is correlated with microbial metabolisms, it could move up on the y-axis and interact with this zooplankton end member. So these are two things that we think might happen, or we have the null hypothesis that zooplankton fecal pellets would remain the same with microbial degradation. So to test this, I'm designing an experiment where we have zooplankton fecal pellets that are being incubated with seawater containing a microbial community. So we were lucky to get fecal pellets. We couldn't go out to sea because of the pandemic, but we have a collaborator, John Steelix, over at the experimental aquaculture hatchery facility that he raises zooplankton to feed to larval fish. So we can take some of the zooplankton and generate fecal pellets like I talked about before. And then we can use those fecal pellets in these incubations to see what the isotope effect is. So I have designed these incubation column containers that rotate like an hourglass. And this ensures that the zooplankton fecal pellets are constantly sinking through bacterial communities and seawater. And we'll measure the fecal pellets at the beginning of the incubation and at the end of the incubation to determine how their isotope signature changes, if at all, during this microbial degradation. So we're going to answer this question, but we also have some interesting findings from our study that were things we weren't looking for that are really cool. And to talk about this, I need to introduce the concept of trophic position and stable isotopes. So the trophic position is really simply the position or level in the food web of an organism or particle. So for example, we have trophic position on the x-axis here. We have phytoplankton, which would be at a trophic position 1 because they're a primary producer. We have zooplankton that would be eating the phytoplankton as a primary consumer or trophic position 2. And we have a fish here that would be consuming the zooplankton and then being a secondary consumer at trophic position 3. So we can determine the trophic position using stable isotopes due to the behavior of 2 groups of amino acids. So delta N15 here is on the y-axis, and our first group of amino acids, the source amino acids in the open boxes, don't have a delta N15 change, or it's not a very large change with trophic position increase. However, we have trophic amino acids, which is a different subset of amino acids that change a constant measurable amount every time we have an increase in trophic position. So using the difference between these 2 sets of amino acids, we can calculate trophic position for an organism or a particle. And when we do this for our zooplankton and their fecal pellets, so here we have all of our various zooplankton and their trophic position on the y-axis, we see that they have really variable relationships between fecal pellet and zooplankton. So zooplankton are in these black diamonds and fecal pellets are in the red stars. So the difference between these 2 is really variable. And when we're piecing together why this would be the case, we're thinking about what a trophic position of a fecal pellet means. So if we have the zooplankton that are eating a trophic position 1 phytoplankton, they will have a trophic position 2 by definition. And then their feces will have a trophic position of 1 to 2 because the feces, the zooplankton cells and metabolites that are in the feces will have a trophic position of 2, and any undigested food will have a trophic position of 1. So if we make a calculation where we subtract the trophic position of fecal pellets from the trophic position of zooplankton, we can get a measure of delta trophic position. And this delta trophic position should be proportional to the amount of undigested food. So we do that for a subset of our zooplankton. Here we have delta trophic position on the y-axis and our various zooplankton here on the x-axis. I have the ball of death, but I'm just going to keep talking. We have our theoretical minimum for delta trophic position, there we go, at 0, and our theoretical maximum of delta trophic position at 1. So 0 would be feces that are entirely digested, and 1 would be feces that are entirely undigested. And when we examine our fecal pellet zooplankton pairs in this space, the first one that stands out to me is these salps from the Northeast Pacific, which are very much at our theoretical maximum. So this means that their fecal pellets contain pretty much entirely undigested organic matter. So it's very similar to what their diet was. In contrast, we have the Sargasso Sea mixed communities, which are at our theoretical minimum of 0, which means that their feces are entirely digested. And we were able to corroborate this salp delta trophic position of 1 with collaborators on the exports project that are making other measurements on those fecal pellets. So we found intact phytoplankton and a lot of genetic material phytoplankton in these fecal pellets. So we're pretty convinced that this is a good indicator that these salps are not digesting their food. And we can draw some hypotheses why our Sargasso mixed community would be at 0 or salps would be at 1. So our salps were around a lot of food at the time, and maybe they don't have to digest all of their food, whereas our Sargasso mixed community doesn't have access to quite as much food, and maybe they need to digest them entirely. Or we could think that if we measure any given organism, they might have a delta trophic position, but if you measure a whole community, you might just get a net zero effect. So a lot of interesting hypotheses to explore with this. I also want to point out the E. superba, which are our Southern Ocean krill, that are also pretty close to our theoretical maximum of 1. So these krill are also taking phytoplankton and eliminating it pretty much intact, which has a lot of impacts on the biological pump in the Southern Ocean. And you can think about this as the krill and the salps are not hungry, but they're still eating phytoplankton and packaging it into larger, denser particles, and therefore exporting it much more quickly than it would have been otherwise. So to summarize the implications of this delta trophic position, we found that some fecal pellets are, in fact, tightly packed, undigested phytoplankton. And in this case, zooplankton are acting as strong exporters of carbon to the deep ocean. And another effect of this is that these fecal pellets, which are full of very high quality food, now being exported to the deep, can become a high quality food source for deep water organisms living deeper in the water column. Oops, I'm sorry. And then our final idea about this delta trophic position calculation is that more work needs to be done to make this method quantitative. So I would say that we probably need to do experiments with aquaria to make sure that this is a quantitative measure of how much undigested food is in fecal pellets. But once that's done, this could be used to further characterize the zooplankton influence on the biological pump. So with that, I want to thank the organizations that funded all this work and highlight the Mary Roche Scholarship, which is funding the incubation experiments that I discussed. I also want to thank my advisor, Hillary Close, and our collaborators, as well as the current and former members of the Close Lab that have had a lot of impact on these projects. So thank you to everyone.
All right, thank you, Shannon. If anybody has any questions, we'll go ahead and move into that portion. Feel free to unmute yourself and ask your question, or if you want to put it in the chat, I'm happy to read it. But first, let's give Shannon a round of applause because that was a great talk.
Hey, Shannon, this is Dennis. That was really beautiful talk and great graphics. You must have taken a long time to prepare this presentation. So
Thank you.
Congratulations, and that looks amazing. So if you had to guess, what's the fate, the disaggregation fate of those fecal pellets if they're digested versus undigested? What's the, how do they package differently? What's their differential fates at depth?
Yeah, I think because we found them in similar ways, so these fecal pellets were all separated out from zooplankton in similar ways. I think they're morphologically very similar, even if they're undigested. But I also think that they may be more labile and therefore more likely to be broken up by microbes if they have a lot of undigested phytoplankton in them. So the disaggregation due to microbial activity might be higher. I don't know. I know that zooplankton have like chemosensors too, so they might be able to tell which particles are tastier than others. So if it has more undigested phytoplankton, they might be able to tell that as well. But yeah, so I think generally they're very similar, but there might be the activity of organisms on them might be different because they're more labile.
So let me ask another question. Is disaggregation the primary fate of fecal pellets, or are they intercepted and consumed? Do you know a primary fate?
It seems like, so based on our estimates of the fecal pellet concentration with depth, at least in Monterey Bay, it looks like they sink quickly enough that they're escaping a lot of breaking up and repackaging. Obviously here in the larger particles, we have a lot of fecal pellets at about 200 meters. And we think it's either because of migration or because this is how deep they would get from night to day. But we obviously have a lot of breaking up of those particles here. So I think for the most part, they make it pretty far, which is interesting.
Thank you.
Yeah.
All right, we've got time for a few more if anybody has any questions.
I'll ask a question if other people need more time to think. Can you talk a little bit more about diel vertical migration and how, I don't know, how would you differentiate between fecal pellets that are expelled at the surface at nighttime and are settling down compared to fecal pellets that are expelled by migrators when they are down there in the mesopelagic?
Okay, so fecal pellets that are, I'm trying to find a figure to point to, but I'll just use this one. So fecal pellets that are made at the surface and sinking versus ones that are made at depth. I think if we characterize what fecal pellets look like as they're microbially degraded, you would expect that fecal pellets that have been sinking for a long time through a soup of microbes would have more of that signature associated with them. So that'd be one way to do it, versus fecal pellets that have been expelled freshly in the deep would have less of that microbial signature associated with them. So that would be one way to do it. If the zooplankton are feeding at depth, the fecal pellets would certainly have a higher trophic position than the ones at the surface where they're consuming mostly phytoplankton too. So that would be another way to do that.
What do you think about, like, I mean, can we get at that by more targeted sampling? So the Monterey Bay stuff was all collected during the day. So, you know, night, day, seasons, do you think that you could end up distinguishing those different effects?
Yeah, so it will, if zooplankton are at the surface, it will take a certain amount of time for even the largest fecal pellet to go to depth. So if you measured particles at depth and separated those as fecal pellets, or you collected zooplankton at depth and incubated them and analyzed their fecal pellets, that would be one way to determine what could be coming from the surface versus coming from the deep. So the zooplankton that we have from the study, I think all of them were collected at the surface during the night, which would mean that they are a mix of the zooplankton that are living at the surface and the zooplankton that are migrating up to the surface. So you could also collect zooplankton that are only at the surface or always at the surface, and zooplankton that are migrating to depths. You could untangle that by measuring various zooplankton at different depths during different times of day. And similarly, you could measure the particles at different depths at different times of day too.
Great, thanks, Shannon. All right, if nobody has any more questions, we'll wrap it up today. Again, a great talk, Shannon. Virtual round of applause.
Thanks.
One day we'll be back in lab.
Mm-hmm.
Might happen sooner. So
yeah.
Thanks, everybody. Have a great Wednesday, and we'll see you on Friday at 11 a.m. for our next Compass seminar from Luna Huron. All right, thanks.
Bye, everybody.